Dear students, so today we're going to be revising meiosis, of which it's the chapter that you've already done in your schools. This belongs to term 1 and it belongs to paper 2 of life sciences. So before I start, I just want to remind you about what you learned about in grade 10, which is mitosis, before I comment on meiosis, because mitosis and meiosis both are cell divisions. Right. So now when we say they are both cell divisions, but they differ in what they do, when we talk about meiosis compared to mitosis, mitosis was a cell division that did not introduce what's so-called genetic variation. Now we're going to be looking at how meiosis introduces genetic variation. What is genetic variation? It is that small difference among the species. Because when you look at us, we're not the same. When you look at your brother, your mother, you may be from the same paternal and the same maternal, but you're not the same. That means there is a genetic variation that has been introduced by meiosis. So now, when I talk about mitosis back then, we said mitosis, what is a cell division that doubles the number of chromosomes to produce two similar daughter cells. Now let's check what is meiosis. Meiosis, it is a cell division. So that's the first thing you must understand. It is a cell division. Now what it does, you learned about chromosomes in grade 10. And you learned that a human being consists of 46 chromosomes. So what is meiosis going to do to the number of chromosomes? It is a cell division that halves the number of chromosomes. Meaning that when I had 46 chromosomes, meiosis is going to halve to 23. So meiosis, it halves the number of chromosomes. It does not double the number of chromosomes. Now, what is the importance of halving the number of chromosomes? We are trying to prevent the doubling effects of chromosomes. As I said, a human being consists of 46 chromosomes. That means when also you as a paternal, you fertilize with your maternal, of which that is your partner, you cannot fertilize 46 and 46. It means that the offspring are going to get 92 chromosomes, which this is an abnormality. So we need to do meiosis so that we have the number of chromosomes. Why do we have to have? So that the maternal can produce 23 and the paternal can produce 23. So that the offspring also will have 46, of which this is normal. So that's the main reason of having the number of chromosomes. We are trying. In a question paper, it can come and say, what is the significance of meiosis? The first thing you must understand, it prevents the doubling effect of chromosomes. Right. Now, the third thing we said is the cell division that halves the number of chromosomes to produce 4 haploid dissimilar daughter cells. So we are producing 4 haploid. Now what does the term haploid mean? When we look at the number of chromosomes, as I'm saying, the normal human being is having 46 chromosomes. This is so to be called diploid. You are diploid. What does this mean? It means you have 23 from your paternal, your father, and you have 23 from your maternal. The 23 is rated as haploid and the other 23 is rated as haploid. That means when this haploid fuse with this haploid in a process called fertilization, that means we're going to produce diploid. It's an N and an N. They're going to give us 2N. So we say this is diploid, this is haploid. So haploid means it's half. When you look at this word, it starts with HA, meaning that it is half of di. Di means 2, half means half of 2, right? So it produces 4, the result of meiosis is going to be 4. Everything that is produced by meiosis is going to be 4. It produced by 4. And whatsoever produced there is going to be haploid, right? Meaning that it's going to be half of what it was. Number 3, it is dissimilar. When I say dissimilar, I mean there is now genetic variation. They have introduced genetic variation. So there are processes that are occurring in meiosis that introduce this genetic variation, of which we're still going to talk about, which is crossing over in prophase 1, random arrangement, random segregation. We're going to look at when we get deep into the process of meiosis. Now, when we look in males and when we look in females, now they undergo meiosis in different ways. Now, the main aim I want you to also understand about meiosis, it is there to produce what is so-called gametes. So what are gametes? Gamete, we're talking about sperm cells, and we're talking about the ovum, of which is so-called egg cell. But you need to use correct terminology because the word O or egg cell is outdated in our syllabus, in our CAPS syllabus. So we're going to use ovum. So this is a sperm cell and this is the ovum, right? So those are so-called gametes. So now we have processes of meiosis. Both, they undergo the process of meiosis. So what is the meiosis that happened in males called? It is called spermatogenesis. So it is called what? Spermato. That means the meiosis occurring in males, it is going to be called spermatogenesis. And what is the meiosis occurring in females going to be called? Obviously, it's going to be oogenesis. Right? So these are two types of meiosis or forms of meiosis. This one occurs in males, it's called spermatogenesis. This one occurs in females, it's called oogenesis. When you look at this word, it says spermato, which means it talks about the sperms. This word, it says oo, of which it refers to the ovum. And then it ends with genesis. The word genesis means formation. So what are we doing here? We are forming sperms. What are we doing here? We're forming an ovum. Oogenesis, formation of an ovum. Spermatogenesis, formation of a sperm cell. Right. Now, we said here we're forming sperms. Here we're forming ovum. So what is the number of chromosomes in each gamete? Whenever you produce sperms as a male in testes, in some area called seminiferous tubules, the sperms must come out having 23 chromosomes, which means it must come out haploid. Right? Why it must come out haploid? So that the ovum, when it brings another 23, which is also haploid, when the sperm and the ovum fuse, that process is called what? Fertilization, right? So that we can produce what is so-called a zygote. What kind of a zygote do we need? Haploid plus haploid is going to give us diploid. That means this zygote is normal. Due to the meiosis that has occurred in males and due to the meiosis occurred in females, it prevented the doubling effect by halving the number of chromosomes. If meiosis did not occur here, we were going to have doubling effect, and then the zygote was going to have 92 chromosomes. That zygote was not even going to even form because it's an abnormal zygote. Now, what are these? What are we talking about when we talk about this? In these 23 chromosomes, it's very important to understand the content of chromosomes. In these 23 chromosomes, going back to 46 chromosomes, the same 46 you're having, we're saying you are diploid. It's a full set, right? Chromosomes, full set. Now, how are they segregated when you go deep? These are chromosomes, but the chromosomes are made up of autosomes and they are made up of gonosomes. So the word chromosome, it can either be an autosome, it can either be a gonosome. Now, what are autosomes? Right. Now, these are somatic chromosomes. Somatic chromosomes. Meaning that you are going to find these chromosomes in the body. Right. They are body chromosomes. They are somatic chromosomes. So the word somatic refers to body. Right. And then we continue. What are gonosomes? Gonosomes, we are talking about sex chromosomes. Right. These are now sex chromosomes. Now, let me make an example about this. I'm saying we have autosomes and gonosomes in this 46. So how many autosomes do you have? You have 44 autosomes. That means how many gonosomes you have? You have 2 gonosomes. Right. That means you have 44 somatic chromosomes and you have 2 sex chromosomes. Now, what are sex chromosomes? Sex chromosomes, in males, we have XY. In females, we have XX. Right. Now, when meiosis, okay, let me explain this. When I say we have 23 here, and I say we have 23 here, this tells me that in this 23, because this 23 is the half of this 46. That means in this 23, I have 22, which was halved from this 44. And I have 1, which was halved from this 2. That means in this 23 chromosomes, I have 22 autosomes and 1 gonosome. Right. Meaning that inside the sperm, it's either the sperm carries X or either the sperm carries Y. But in an ovum is only subjected in carrying X because there is no other alternative gonosome. They only have 2 X's. Right? That means if a male is carrying X in its sperm, that means when X combined with its X, we're going to get an offspring, which is a female. Right? But when a male produces sperm containing Y, when this Y fuses with that X, we're going to get an offspring, which is XY. Right? Sometimes there can be a case whereby we say the male failed to separate XY. That means when we say the male fails to separate XY, we are going to get a sperm containing XY like this. Right? Only to find that the mother is going to produce a normal gamete, the mother is going to give us X. And then we get an offspring which is going to be XXY. Of which this is now abnormal. It's called Klinefelter syndrome. This is a male with a feminine body. Why? That's because there's been an extra X chromosomes due to chromosomes failing to separate. Right. So now I want us to continue and look at, because this is still introduction, it's very important to understand this thing before you even go inside the process of meiosis. Right. Okay, let me just wipe this thing out and then we are going to continue with our lesson. All right. Now, when we talk about meiosis, it comes into 2 phases. So when we look at meiosis, it is in 2 phases. So we have meiosis 1 and we have meiosis 2. Now, when you look very closely to meiosis 1, meiosis 1 is more like mitosis that was learned in grade 10. Just that there are things that are occurring in meiosis 1 which were not occurring in mitosis. So now, the phases of meiosis, we have a phase which is called an interphase, followed by prophase, followed by metaphase, followed by anaphase, and lastly anaphase, and lastly telophase. So we have these phases, Right, but I'm sure you have an idea that interface is not regarded as a phase on its own. Why? Because in interphase, this is where we get a process which is called DNA replication, whereby the chromatin network condenses and unwinds to form chromosomes. Right. Now, looking at what is happening here, to give you a hint of how to see prophase. Prophase start with P. You must know that we are still doing what? Preparation for the whole cell division, right? Metaphase start with M. You must know that there are gonna be chromosomes lying in the middle, of which we call the equator. Anaphase start with A. That means there are chromosomes that are gonna be moving apart from each other, right? And then telophase start with a T. That means we have reached the terminal phase. We have reached the end of our cell division, right? So now, there is one thing that is important, structures. Before you even start the phases, structures. Now, what are the structures are we referring to? The first one is going to be this. So this is so known as what? A chromosome. Right. So this is a chromosome. What forms the chromosome? Chromosome is formed by 1 and 2. What is this 1 and 2? When you label them, you call them chromatids. So these are chromatids, meaning that a chromosome is formed by 2 chromatids. Now, what joins chromatid 1 and chromatid 2? They are joined by what is so-called centromere. Right. So these are structures. Not all the structures I'm referring to, but it's very important to understand the structures because we're going to be using diagrams when we go further with our process. So this is a chromosome. Chromosome is formed by 2 chromatids that are joined by the centromere. Now, another thing you will see what is so-called a centriole. Centriole produces what is so-called spindle fibers. Right. So the centriole produces the spindle fibers. So that's a centriole. Now a question will say, where is this centriole coming from? Now the centriole comes from what is so-called centrosome. So you have a centrosome. When they indicate a centrosome, you're going to see like this, something like this. So the big thing, this one, it is the centrosome. Inside the centrosome, we have 2 centrioles. So when the centrosome splits, it is going to produce 2 centrioles that are going to go in opposite poles. So we have a centriole in this pole, we have a centriole in this pole. Where are they coming from? They're coming from the centrosome. So when a centriole arrives, it arrives with the spindle fibers. What are the spindle fibers going to do? The spindle fibers are designed to hold the chromosomes in position. So when the chromosomes are lying along the equator, they are held by the spindle fibers. Right. And then when you see chromosomes like this is a chromosome and this is another chromosome. When you see them in this form of pairs, we call them homologous pairs of chromosomes. Right, we call them homologous pairs of chromosomes when they are in this form. This is not a homologous pair. This is only a homologous pair. Now, a question will come and say, why are they regarded as being homologous? Right? What are the characteristics of homologous chromosomes? Now, when you look at the word homo means the same. Right? That means when we call them homologous chromosomes, we mean they have same structure, similar length, similar size. You understand? Those are the characteristics of homologous chromosomes. They have the same structure, same size, same length. Do you understand? And also, if they are positioning for a gene, it will be positioned in an alternative position, which is called an allele. The position will be the same. So in that manner, we say they are homologous. Understand? So we only call them homologous if they have the same size, same structure, same length, and the same position of a gene. All right. Now let me start the process now, because clearly we have an understanding. Interphase. I'm starting here. I said to you, this is not a phase. You don't call it a phase, right? Now, interphase. How do you see that this is interphase? Now, there is one thing I forgot to say. Meiosis occurs in the nucleus, and the nucleus is inside the cell, right? So, you must indicate the cell membrane. You must indicate what? The cell membrane. And then, inside the cell membrane, you now have to indicate the nucleus. So if this is the cell membrane, then this is the cytoplasm. Right? We're still looking at the cell. We haven't moved inside the nucleus. And then this is going to be the nucleus. So this is now not cell membrane, because this is not a cell. It is now nuclear membrane. Is it clear? If it's a cell, you call it cell membrane. If it's a nucleus, you call it nuclear membrane. Right. Now, this was the cytoplasm. The word cyto means cell. So, this is not a cell. So, this is not going to be the cytoplasm, but it's going to be the nucleoplasm. It's going to be the nucleoplasm. It's the plasm of the nucleus. And then we said this is the cell, this is the nucleus, and then we have the nucleus of the nucleus. The nucleus of the nucleus is called the nucleolus. It is called nucleolus. So this is a nucleus of the nucleus. The bigger one is the nucleus, this one, the one I'm highlighting with red. That's the nucleus. And this one, it is the nucleolus. Right? Now, in the nucleus, what are we going to find? We are going to find capillaries of networks. And these capillaries of network are called chromatin network. They're called what? Chromatin network. Now, we want to start our prophase, but the problem is we do not have chromosomes. How are we going to form chromosomes? Who's going to help us form chromosomes? It is the chromatin network. By which process? There is going to be a process occurring here, which is DNA replication. So what is DNA replication? This is whereby the DNA duplicates itself to make an exact copy. So the chromatin network, as you can see, it is a network, right? So what must happen to it? It must unwind, right? It must unwind. Why it is unwinding? So to form a chromosome, right? So it is going to unwind under the process of DNA replication, and then it is going to form a chromosome. Then in the next phase, which is prophase, that is where you're going to start to see chromosomes. But in this phase, that's why it is not regarded as a phase, because it is still also, it falls under a preparation. We're still trying to get everything together before we start. So this is going to be our chromatin network. It unwinds during DNA replication to form chromosomes, right? And then we go to prophase 1. I said meiosis come in 2 phases. Phase 1 and phase 2. So I'm still looking at phase 1. So at prophase 1. How do you see prophase 1? What happens in prophase 1? I'm going to draw this one, of which already you know that this is cell membrane. And then I'm going to draw this one, of which already you know it's a nuclear membrane. And then I'm going to draw this one, of which already you know it's a nucleolus. But now, the difference, look at the nuclear membrane and the nucleolus in interface. They are not drawn in broken lines. But in prophase, they are drawn in broken lines. Why are they drawn in broken lines? To indicate that they start to disappear. So in prophase 1, the nuclear membrane and the nucleolus, they start to disappear. Meaning that in the next phase, we won't be seeing nuclear membrane and nucleolus, but we will be all in the cytoplasm. Is it clear? All right. And then, now you're going to see chromosomes. Where are these chromosomes coming from? They were formed by chromatin network unwinding during DNA replication. Now, there is a process that occurs here. So you see chromosomes, you see 4 chromosomes. So the first thing I said the nuclear membrane and the nucleolus disappear. That's the first thing you have to say. Nuclear membrane, nucleolus disappear. What else do you have to say? You are going to see chromosomes like this. And you ask yourself, what happened? Why chromosomes are having mixture of colors? Why the chromosomes are having mixture of colors? It is due to the process of what? Crossing over. Right. That's the main process that introduces genetic variation. Remember, I said there is so-called genetic variation, and I said it is the difference among the species. Now let us check how crossing over occurs. Let me just say, this is paternal, the father, and the father, this is the father's chromosome. And this is maternal. And this is going to be the maternal chromosome. Now, if you look at this, these are 2 homologous chromosomes overlapping at each other. So, where they overlap, where they meet, this point is called chiasma. So, now it's only the chromatids of the homologous chromosomes that overlap to each other. Why are they overlapping? Now, the father is blue, the mother is black. I'm making an example. But we do not want our offspring to take one side. We want the offspring to have a mixture of both parents. So what is going to happen? This blue is going to have a small portion. When you draw blue, you're now going to draw blue in this way. It's still the same chromosome, but the problem is it's now having a mixture from the mother. And when you draw black, this is how you're going to draw black. Black is now having a mixture from the father. That means the offspring is going to get these chromosomes having mixture of both parents. That's what we say, the importance of this process. It does what? It introduces genetic variation. So other textbooks will say it introduces genetic diversity. So That is the process. That is one important process occurring in prophase 1. So now, it only occurs in prophase 1. All the phases that follow, they are going to go with the mixed chromosomes. There is no new crossing over that is going to occur. So I said prophase preparation. So how many chromosomes do we have here? We have 4 chromosomes. We're now going to metaphase. And I said in metaphase, chromosomes will be lying along the middle. Right. Let me just wipe here. So we're now moving from prophase 1 to metaphase 1. Now, when we say metaphase 1, we said this M tells us that the chromosomes will be lying along the middle. Right? Now, lying in the middle we're talking about is the equator. Some other textbooks, they call it epiphyseal plate, other call it equatorial plate, other call it metaphyseal plate. But for us to master, we're going to say equator. Right? So now, never forget this. I always emphasize students that never forget the cell membrane. First, to draw the cell membrane. Now, what are you going to see here? Let me just draw here. I forgot to add something here. In prophase, you're going to see them putting something like this. And they want to speak about it. Why? This is what I said, it's a centrosome. The centrosome now needs to split so to release these 2 centrioles. So that's what first happens here. The centrosome splits. That centrosome. It splits to release 2 centrioles. So we now have centrioles, right? And then I said centrioles, they produce spindle fibers. Now you're going to see spindle fibers being produced. And then I said the spindle fibers hold the chromosomes in position. Since we have 4 chromosomes, they are going to be arranged along the equator in this form. Now, when we get back to what we talked about in structures, we said this arrangement is homologous pairs. Right. So when they say explain what happened in metaphase, now there is one thing that has the similar importance as crossing over. We said the importance of crossing over was to introduce genetic variation. What's the next thing that occurs here in metaphase that is doing the same thing? That is so-called random arrangement. So the random arrangement has the same significance with crossing over. They both introduce genetic variation. Right. Now, when they say explain, because this is revision, we're just reminding one another. We won't be doing deep of what you've learned already. When they say explain what happens on metaphase, now you have to say chromosomes randomly arrange themselves along the equator in what? In homologous pairs. So the arrangement of metaphase 1 will occur in homologous pairs. Therefore, these chromosomes, they randomly, there is no selective force that is going to arrange them. They randomly arrange. Never forget that all your chromosomes are having the mixture. What caused them to be like this? It is the crossing over that occurred in prophase 1. So that's what happens in metaphase. Chromosomes randomly arranged along the equator in what pair? In homologous pairs. And then we move from metaphase, right? We go to anaphase. Now I said anaphase it tells us the chromosomes will be moving apart. Now, if you look at the structure above, you see that the chromosomes are held by spindle fibers. The only thing that is capable of pulling the chromosomes apart, it's the spindle fibers. Since this one is attached to this one is attached to this one is attached to this one, and this one to this one. Therefore, this line will pull this chromosome. This line will pull that chromosome. This line will pull the chromosome down, and that one down. So they go in opposite direction. So chromosomes are pulled apart by the spindle fibers. So you're gonna see in anaphase. Never forget about this one. You're gonna see in anaphase chromosomes have separated. They are no longer homologous. You can't call something that is so separated like this a homologous. No. So when they say explain what happened in anaphase, these are spindle fibers. And the only thing that is capable of pulling the chromosomes are the spindle fibers. So what happens here? Spindle fibers contracts and they pull the chromosomes in opposite poles, or they pull chromosomes apart from each other. So the chromosomes are pulled apart. These two is going up, the other one is going down. Now, I once explained earlier when I introduced the issue of this. I said this is due to maybe the father failed to separate XY. The separation, where is it failing? It only fails in anaphase, because this is where separation occurs. It cannot fail in metaphase. It cannot fail. It only fails here because this is where we get what is so-called random segregation. So the chromosomes segregate. They move apart from each other. Now, I want you to look at these three things. You have crossing over in prophase 1. You have random arrangement in prophase 2. Sorry, in metaphase 1. And you have random segregation in anaphase 1. How are all these three processes important? They all introduce genetic variation. So when the question comes and say, name three things that occur in the phase of meiosis which introduce genetic variation, you say in prophase 1 we have crossing over, in metaphase 1 we have random arrangement, in anaphase 1 we have random segregation. So in wrapping anaphase we're saying spindle fibers contract and pull chromosomes in opposite poles. So now we're going to the last phase, and our last phase is going to be telophase. Now I want you to look at I want you to notice something about telophase. I'm first going to draw telophase. Now, how do we get something like this? We said that this one goes that direction, and this one goes this direction. Meaning that cell A is the other portion, cell B is the downward portion. Now, the only problem I want you to see there, the whole cell have divided. It was not divided, it was not divided, it was not divided in prophase, but it has divided here. Meaning that the cytoplasm have divided. And what do we call the division of the cytoplasm? It is called cytokinesis. So the cytokinesis has occurred. There was a division of cytoplasm. Now, of which makes you to see two cells. So we're now having cell A and cell B. Right? Now, another thing, there is one thing I said in prophase 1, it disappeared, but now it's back. Nuclear membrane, nucleolus. So there is gonna be nuclear membrane and nucleolus reappearing in telophase 1. So we are not done about the whole meiosis structure. Because we said when we defined it, we said it is a cell division that produces 4 haploid. This is not 4, this is 2. So we still need to undergo meiosis 2, of which is going to be the simplest because you are going to be talking as you were talking in meiosis 1. This is telophase 1. I'm now going to start in prophase 2. We no longer start in interphase 2. We do not have interphase 2 because interphase is only a preparation whereby we're forming chromosomes. We already have chromosomes, so we don't need interphase. Right? And then prophase. I'm going to take one cell for now. I'm going to take one cell. I'm going to take this cell from telophase and put it in prophase. The moment I put the cell in prophase, this is what happens. Don't forget this one. I'm going to take the cell, this one. Immediately I put it in prophase, the nuclear membrane and nucleolus start to do what? To disappear. How many chromosomes do I have? 2. Do you understand? That's the only thing you have to say about prophase 2. The nuclear membrane, nucleolus. I just took this, only cell A, and put it under prophase 2. I'm still dealing with cell A. I take cell A and put it under prophase 2. The nuclear membrane starts to disappear. The nucleolus starts to disappear. We go to metaphase 2. We no longer have 4 chromosomes. That means we cannot have homologous chromosomes or homologous arrangement. What is going to happen? This is what is going to happen. Can you see the difference between that metaphase and this metaphase? In this one, we are arranging in a single row. So whatever you said in metaphase, you said in metaphase, chromosomes randomly arranged along the equator in homologous pairs. But now you have to change that and say chromosomes randomly arrange themselves along the equator in a single row, not in homologous pairs. So once you see chromosomes be arranged in a single row, that's where you know it all. I'm now dealing with anaphase or metaphase 2. And then we go to, from metaphase, we go to anaphase 2. Ana, A, apart. Now, I want you to look clearly. There is no other way we can separate the chromosomes if we are not separating the chromatids. Right? So, what is going to happen in anaphase? We are now going to have chromatids, right? When you join this, it forms that. But now we have chromatids. So whatever you said in anaphase 2, you said in anaphase 1, the spindle fibers contract to pull chromosomes, right? But now it's not, we're not pulling chromosomes. Spindle fibers contract to pull chromatids. That's the difference between 1 and 2. In the other one, we're pulling chromosomes. This side, we're pulling chromatids. It's clear? All right. And then we move to telophase 2. Still the same thing. This one goes that side, this one goes this side. And then we're going to have the process of cytokinesis. So I'm going to have something like this. Cytokinesis, nuclear membrane reappears, nucleolus reappears. Nuclear membrane reappears, nucleolus reappears. Two chromatids. Right. Mind you, I was still dealing with cell A. I'm now dealing with cell B. I take cell B, I do the same process. I put it under prophase 2, metaphase 2, anaphase 2. It will give me the results similar to cell A. Is it clear? And then I put the result of cell B, chromatid, nuclear membrane. Now I have a 4 haploid daughter cell. And this is going to be the end of meiosis in that case. Because we have achieved what we said. Right? Now, students intend to lose this mark. A question will come and say, tabulate between. Once they say tabulate, you need a table, because once you write your own thing, even if you do not know what to write, just write the table. There is a mark for writing a table and leaving it like this. An empty table, there's a mark. Because they'll say tabulate 4 differences. 4 differences, you're going to be 1, 2, 3, 4. And then they say 5 marks. What's the other one for? The table. So if you did not write the table, you have lost a mark for the table. You can just write the table and leave it like that if you don't know. All right. Now, they are saying tabulate between meiosis 1 and meiosis 2. Do not jump. Go phase by phase. Start here. In prophase 1, there is process of crossing over. In prophase 2, there is no crossing over. In metaphase 1, chromosomes arrange along the equator in homologous pairs. In metaphase 2, chromosomes arrange along the equator in a single row. In anaphase 1, spindle fibers pull chromosomes. In anaphase 2, spindle fibers pull chromatids. In telophase 1, 2 daughter cells. In telophase 2, 4 daughter cells. Do not jump. Go phase by phase. Right. Now, there is one thing I also want to emphasize before I close the session of meiosis. The issue of karyotype. One is going to ask, what is karyotype? Why is karyotype important? Now, I talked about, when I introduced, I said a person is having 46 chromosomes. In this 46, you have 44 autosomes, 2 gonosomes. Now, how are those chromosomes arranged? You're going to see a diagram like this. And let me just say this. The 46 chromosomes you have, when you group them in pairs, you get 23 pairs. Meaning that chromosomes are always supposed to go in pairs. So you're going to have pair number 1, 2 chromosomes, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23. And then I say XX, 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 XX. So these are your chromosomes going in pairs, of which there is normal. All right. And then there's something I said. I said you have 44 autosomes. Meaning that when you group them in pairs, you are going to take pair number 1 up to pair number 22. So pair number 1 to 22, those are autosomes. And I said you have 2 gonosomes, right? Gonosomes, pair number 23. So this is where you are going to see the gender in the karyotype. You're going to see the gender in karyotype in chromosome number 23. If it's like this means this karyotype is of a female. If it is like this is male. They will not give you this. They will not write like this because it's going to be obvious. They'll give you something like this or something like this. This is, you can see like an X. No, it's not X. It represents Y. If it was representing X, they were going to be the same size to show that they are homologous. So this is Y. This is also representing who? Y. Y is represented as a short chromosome. It is because the Y chromosome is always weak. It is the weakest chromosome. Right? It is the weakest chromosome. That's why in genetics you find that the Y chromosome cannot carry a sex-linked disorder. It is because it is very weak. Right? So it's very important to understand the issue of the karyotype. So now, there is this karyotype, pair number 21. When the chromosomes can you see that this chromosome failed to separate? And the failure of chromosome to separate is called non-disjunction. They failed to separate. It is called non-disjunction. How is this non-disjunction going to affect the karyotype? It will result to an extra, because if you look at this, we are short here, and we have an extra one here. So it's going to go to karyotype number 21, and it adds the dead chromosome. That means when you have 3 pairs of chromosomes supposed to be having 2, this is now called trisomy. Tri means 3. So you are now having 3 chromosomes instead of 2. And that is going to give you a disorder which is called Down syndrome. You are now having 47 chromosomes instead of 46. That means you are now having 45 autosomes instead of 44. Right? So it is very important to do question papers after the session. After you have done the session, you need to go through question papers, past question papers, because it is not wise to know the theory parts without the practical parts. Right? So go and practice more questions. Ask questions, refer to your notes, go back to your notes, play the video, refer to the video, replay, rewind, forward, do you understand? So you can understand whatever that happens in Meiosis chapter. So thank you very much for attending today's class. See you next time.